So in addition to Peggy and Chris, we have several other SCI experts in the audience, and we'd like to give them the opportunity to comment on the work we've just talked about before we open the floor to questions. So I'll introduce each speaker, call them up to the front, and let them take the floor for a 2-minute comment. I think I'm just going to call everybody up and then ask them to speak all together. Hopefully that will save some time. So, already almost up here, Dr. Wolfram Tetzlaff is the IPOR director and a UBC professor in the Departments of Zoology and Surgery. Thanks, Dr. Tetzlaff. Dr. Lyn Jakeman is the program director at the National Institute of Neurological Disorders and Stroke and was previously a professor at the Ohio State University Wexner Medical Center. Dr. Andrei Krassioukov is a staff physician here at GF Strong and a principal investigator at IPOR, where he is the director of the Autonomic Research Unit. Dr. Chris McBride is the executive director of SCI. She didn't get the memo. Dr. Brian Kwon is a spine surgeon at Vancouver General Hospital and the Canada Research Chair in Spinal Cord Injury and a principal investigator at IPOR. So we're going to create a very awkward 2-mic situation so that our online participants can hear. And I have to be the heavy. 2 minutes. 2 minutes, folks. So when you see me wave my hand around, that means pass the mic along. Well, wrap up your comments. Who has the...
Good evening, everybody. Thank you for coming. I have to surprise Jocelyn now because I think Peggy summarized it so nicely. I would rather save the time for the questions from the audience at this moment.
Oh, nicely done.
I don't know if I can follow that.
Can you just leave that for while you're talking? That's just the microphone right here.
Okay. Thank you. I'm getting my AV instructions. So only 2 minutes, and what I wanted to point out were a couple of things. One is that all of, both of the dramatic examples that the speakers spoke of today were both dramatic examples of things that were surprising findings, but based on years and years and years of basic research. And they, in both cases, were surprising findings, which I think changes the way we look at research a little bit now, because I think that in both cases, we can ask a lot of questions about how it works, but we've certainly reached a point where we may be able to enable function in a very, very few number of fibers, which is something that we didn't think was really tremendous in humans before.
Good evening, and thank you all for coming.
This is your transmitter.
That's my transmitter.
So hold on, that's for the online.
It's like my epidural stimulation.
Exactly.
I think that these studies really highlight, I guess, the culmination of a lot of research that I think encourages us in the research field to keep going and encourages, I think, a lot of the community around the progress that's being made. I think that it is worth pointing out, or at least reflecting on the fact that it has been, decades of work that actually culminated in these research papers, and that provides a bit of a lens around how progress in science really occurs. And because I think when people look at these, it's easy to forget that they were many years and in some cases decades in the making. I do think that as a spine surgeon that treats mainly acute spinal cord injuries, I think that particularly the epidural stimulation work really suggests that the things we do in the very acute time frame to try to minimize damage and maximize function may be really relevant if it is indeed these very few pathways in the spinal cord that are mediating this kind of function that can be then kind of woken up with this type of technology. So, when I look at it as a treatment that's applied in a more chronic setting, based on what we think may be happening, it certainly makes me feel very encouraged about the kind of work that we do in the acute setting, where we're really trying to maximize the amount of tissue and maximize the amount of spinal cord that may be available for this as a substrate for this type of intervention. And now for Dr. Krassioukov.
I also, I believe, fortunate that Dr. West did an incredible job describing events, what's happened with epidural stimulation in Louisville, Kentucky. And I only would like to add that I'm very fortunate, and my team very fortunate, to join colleagues in Louisville. And particularly, I would like to acknowledge the NIH, actually, which sponsored First for Patients in this research. But particularly, I believe, all team here in Vancouver and all team in Louisville, Kentucky, would like to acknowledge the courage of First for Patients with spinal cord injury who agreed to participate in this research, because we do appreciate the fact that every single patient who made decision to go through the clinical trials, doesn't matter if it will be non-invasive clinical trials or invasive clinical trials, it is a big commitment for every single individual with spinal cord injury to give their time, their commitments to work with a clinician and scientist in a very, very important issues where we definitely still have to deal with many, many, many aspects of uncertainty, where we would like to make big progress. We would like to cure paralysis. We would like to cure bowel, bladder, cardiovascular dysfunctions. And as you see, we're doing progress. There are some hope, but we still have many, many, many questions to answer. As you saw today, what our junior colleagues described to us, there are many, many questions that we have to answer. And again, I would like to say thank you to our patients, to our participants in clinical trials. We need your help, we need your collaborations, and that's what we're here for.
Thanks, Andrei. So I guess as someone who spent a good part of his career in the research world, and now who works for a provincial organization that serves people with spinal cord injury, helping them to adjust, adapt, and thrive in their lives every day, I think it's really important to remember that what makes a scientist really excited isn't always something that's going to make people with spinal cord injury really excited. And a small but important incremental finding in research is really exciting when you're a scientist. But it can, that excitement can get translated into a hope that can be misinterpreted by media all around the world to make everybody think that the science is advancing much quicker than it is. And I think the point about the fact that the science that was presented today, well, I'll say some of it was science, is the culmination of maybe a decade or more of work. Well, I think what you're seeing is another point along that long road. And where we'll be in 10 years from now, people look back and say, well, that was a really neat incremental piece that led to a whole bunch of new research that is another incremental step towards something bigger. So I think the context for this is really important, and the perspective is really important.
I'd like to congratulate them all for staying under 2 minutes. My hands are now full. Okay. So thank you all. We really appreciate having the opportunity to hear from up-and-coming SCI researchers and also experts in the field. So we've got, I hear, more than 20 online on the internet watching, and a full house here in the room at GF Strong. So I'd like to open the floor to questions. I think the easiest way to do this is to raise your hand if you can, and if not, nudge the person next to you and ask them to raise their hand for you. And we're gonna try and alternate, you know, every couple of questions, we'll take one from online as well. So when Fernando raises his hand, we'll know. So are there any questions? Okay. Can someone, one of the organizers, can you guys do the, like, picking for me? Because I can't really see them.
Oh, I have two questions.
Might not work.
No, no.
Okay.
That's right.
Here, so you wanna just help pick the question?
So still more. The question will only be for this half.
This is a simple question from somebody undergoing it. Can somebody up there explain what nerve buzz is? What does it mean?
What is nerve buzz? What does it mean? Anyone want to take that?
What causes it? Where is it going?
What causes it and where is it going? Is that a,
like the feeling that you've got like pins and needles in your hands. Well, so the question was about what the nature of nerve buzz is, and I think what just so we're all relatively the same page, what we're talking about is the sensation of sort of a pins and needles feeling in part of your body. This is a very common feature of spinal cord injury and other nerve types of injury. And I see a lot in my patients that have been relatively simple degenerative conditions like a slip disc or something in their neck or back. But in the context of central nervous system injury or spinal cord injury, the exact cause of this sort of ongoing pins and needles or sensory inflammation that's unwanted or undesirable or unprovoked is not totally understood. We think that it relates to some ongoing activity and stimulation in the spinal cord that is not that is a result of some aspects of the nervous tissue being damaged that allows other nerve tissue to be activated, or the spinal cord is a very complex organ where certain activities actually suppress the activities of other nerves, and when you influence that or affect that balance, then it can lead to these altered sensations and whatnot. And so, unfortunately, our understanding of exactly what is causing that, and in particular how to treat it, is well, as you can probably tell, rather infancy. Professor, do you have anything else to add?
Totally agree with you. It is a component what we call neuropathic pain, and it's commonly we see with a central nervous system damage, such as spinal cord injury, stroke, or with a peripheral nerve injury, when nerve root compress or peripheral neuropathy occur in diabetes. For example, one of the common people with diabetes will suffer with the same nerve bus injury. That's right. And then, unfortunately, we are very limited with number of medications that we can offer people with this kind of continuous nerve pinching sensation, burning sensation, pin and needle sensation. One of the most common drugs is gabapentin, neurontin, or presently Lyrica in other medications. And unfortunately, all these medications commonly have side effects. And people who can tolerate these medications crave, and most commonly, unfortunately, all people describe them like heaviness, fatigue, sleepiness, and this is difficult to treat, one of the most difficult to treat kind of pain.
thank you. So we have another question over there. Dave? Well, let's start with one.
No, we like the first one.
I'll come back to you for the second.
That's one. Now, I can ask you,
one is just has to do with chronic injuries. I mean, I was injured 40 years ago. I think, and I'm more involved in quality of life issues and community development and that sort of thing. I haven't been to a research meeting in many years. I was focused on that in the beginning. It was all about cure, and now it's funny that I don't really, quality of life, I don't really pursue that. But I'm kind of curious what is going on with chronic injuries like mine. I just, in regards to the epidural stimulation, I was curious, you said that you don't really know what's causing these results, but going into the research and getting approval for the clinical trials, there must have been some theory about what was thought was going to happen. And I'm wondering if the results are in conflict with the theory to do the research.
Good question, dude.
We have a solution to cords on the floor. That's my question. Yeah, you?
Should we give answer?
Yeah.
Give possibility
Chris,
to
you
answer
want it?
junior scientists for
Let's just put him on the
Dr.
spot.
West.
Come on, junior scientist.
You want a faculty appointment.
Okay, I guess I'll try and take that one. So right, I mean, there's actually 2 or 3. So I think, as the panel up here alluded to, kind of 10 to 15 years of animal work has preceded some of the stuff that was done in humans, and there's certainly some theories that are put forth from some of the basic science literature, mostly done in rodents, but also done in rabbits and cats as well. And I guess this kind of goes back 10 years, and really there's 2 theories, at least as my understanding. So one is this idea that even in somebody that is told they have a motor and sensory complete injury, there is some possibility that anatomically, if we were able to take the spinal cord out and look at the spinal cord, we might find evidence that there are still some pathways that are intact. Now, the way in which we assess this clinically, it's almost impossible, even with imaging techniques in the clinic at the moment, to see in that much detail in the human. So one of the theories is that if you have a small amount of information that can still travel down the spinal cord, but usually that would be insufficient to cause the muscles to contract, if we can effectively boost that amount of information by putting in this epidural stimulator at the base of the spinal cord, then we can create an environment where there is suddenly enough information for it to cause movement. Now, that's really for trying to understand how people can voluntarily move after this. There must be some way where those fibers that previously went from the brain to tell the toe to move, there's still some information getting through in that area. And really some of the other evidence out there is that we also know now that the spinal cord can reorganize quite a lot after injury, and it's potential that some of the pathways that were previously involved in movement, or some of the previous pathways that might have been involved in sensation or smell, for instance, some of those pathways might change their function a little after spinal cord injury. There might be some reorganization, and so there might be some way that the information can now be still moved down the spinal cord to elicit movement with the presence of that epidural stimulation. I should say that they repeated all of these experiments without the epidural stimulator on, and none of the participants had any voluntary movement. And certainly, yeah, that's probably my best attempt at that. So, anyone want to say more?
You ask a very important question. When is the threshold when scientists who made some interesting discovery, and then we will make decision, it's time to move to clinical trials? That's what we do have in the science and clinical practice, a very strong regulation, bodies, ethical boards, which looking after this kind of transitions, when we do have to show evidence that we have enough information, threshold, that there is enough data, enough evidence that we are ready for clinical trials. But there is right now more and more tendency in research that, for example, people with spinal cord injury or people in the community could actually influence decisions of these boards, then where we somewhat in a fence and decision could be pushed further. For example, with epidural stimulation, what's happened, Dr. West is absolutely correct. We had years and years of data from Dr. Reggie Edgerton laboratories, Dr. Hartman Susan laboratory with animal research. Then this gave us understanding we do have some kind of base information that it could work. But then money came on board. Who will fund, who will sponsor this data? And we got first grant from NIH to support first 4 patients. But then, as Dr. West said, $30,000 is one grant. And then $30,000 is care for implanted patients. It's extremely expensive enterprise. Next step, Christopher Reeve Paralysis Foundation Board of Directors stood up and said, we want it right now into the wide clinical practice. And Board of Directors of Christopher and Daniel Paralysis made decision. We will fund next step of research. We have right now good preliminary data from an NIH trial. We will fund next step. And that's what Dr. West mentioned. Christopher Reeve and Daniel Paralysis Foundation made a target $12 million to fund this research. That's why this is a question you asked, very important.
Thank you both. If you're interested in more information about that, if you go to, I think it's Christopher Reeve Big Idea on the web, they have lots of videos on it as well. So we have a question online. I'm just going to take a web question first.
So the question is about epidural stimulation. What is the best way to optimize epidural stimulation more efficiently? And how to find the best stimulation signal, amplitude, or pattern and location more adaptively and smartly?
I think that's Dr. Mark's.
Okay, Chris.
Yeah. Thanks, Chris.
Okay, so yeah, that's a really good question, and I think as I mentioned in my talk, the technology at the moment is pretty limited. Actually, the way in which this all came about was from a history of being in pain research and individuals that suffer from chronic pain being implanted to try and help with that. And so really they've taken that same approach and now are trying to apply it to spinal cord injury. With the current setup that we have, you're able to change only very few stimulation parameters. So you can change whether you're stimulating high, medium, or low within that array. You can change the frequency or the intensity of that stimulation. But I mean, in the future, it would be really neat to see something where if we can develop the appropriate software, where perhaps you could have some kind of intelligent stimulation that is taking some of the cues from the environment that are coming back into the spinal cord and developing the appropriate movement out the other side. And I know that's a very big area of research and something that Dr. Hartkemer has been really interested in, and we're actually trying to put it out there for people to try and help and have ideas. IBM and people like that are also trying to help with this and try and find a better way where we can optimize the parameters to either cover a wider part of the spinal cord or to, as I say, to try and do this intelligent type simulation.
Just to add, Reggie Edgerton has actually started to do some patients with just transcutaneous stimulation. And in some people it actually works. And it's really a bit of a trial and error. You cannot really well predict who is going to respond to and who is not. Other people like Dr. Vivian Moshova, who is speaking tomorrow on Wednesday at IPAD at the annual meeting, she is doing intraneural stimulation, intraspinal cord, I should say, stimulation with a multiple needle array, which in itself has other questions, whether it's damaging and has other side effects. So really the world is still open where to get the optimal approach and what to actually try in a human, and every human injury is different. What was really guiding this research was our insight over the last 2 decades that the spinal cord itself can actually learn. And if you take a cat completely transect the cervical spinal cord, put it on a treadmill, and stimulate a little bit, and just stabilize balance. The cat actually can learn how to step. So those patterns are ingrained. It's just a matter of triggering those type of motions. So the spinal cord itself is actually a highly plastic piece of the nervous system itself. So our thinking of it being just an execution of our brain commands has now been greatly modified. And it's now becoming subject of the next decade to figure out what is the most effective way. And every person, every patient is different in that regard.
There's also video of Sue Harkema's talk from a year ago and Reggie Ederson's talk at a recent conference. We can put it on our SCI blog at icord.org. So if you're interested in further viewing on that topic, just check there tomorrow. I want to take some questions from this side of the room. So who's going to read? Okay. Heidi's going to read them.
So we have a question on paper here. Mike's still working? Okay. So the question is, did we just see what is on the leading edge of stem cell research to treat spinal cord injuries? So I'm guessing that means the study that we just covered, is that kind of the most exciting thing that's happening right now? And so that's a really good question, and I'm sure Wolf will be able to jump in on that as well. But there's actually a lot of really exciting work that's being done at the experimental stage. And so looking at rodent models and looking at different cell types, because there's different cell types that could be potentially beneficial in some situations and not others. So I would actually argue that there's some really exciting stuff that is going on at kind of the preclinical stage. Now, with that being said, there are some clinical trials that are going on, and I'll probably refer to Wolf on this because he knows this stuff a little bit better than I do. And he can talk to you a little bit about some of the work that's going on right now.
So at the moment, there are about a handful of trials spearheaded by different companies. For example, there are neural stem cells, which are lines that we're taking from embryonic spinal cords, and two companies, NeuralStem and the other one is Stem Cell Inc., are pushing trials. Stem Cell Inc. has already given now to 5 patients in Switzerland these type of cells, and these studies so far have been given to people with thoracic injuries, and some sensory changes have been observed. It's too early to actually make any conclusions here. NeuralStem has not yet started. They will start with a different line. And it's all very, at the moment, very premature to draw to conclusions. But what we can say so far, it looks safe to put cells into human spinal cords as far as we can tell. Based on that small n. We haven't seen tumors yet. It may take more time to actually assess those kind of risks, which is necessary. Every stem cell has the risk that it could actually bear a tumor formation in the patients. We saw when Geron was trying their trial with their cells, which was an oligodendrocyte precursor cell line. The trial was stopped for financial reasons, that they were put under the burden of following up every individual patient for many, many years to rule out tumor formation, which was highly expensive. I think they needed to budget about $487,000. And this is when the very much by stock market driven executives said this is too expensive, we don't want to go there. Unfortunately, the preclinical data to the Geron cells were not that strong. And that trial was heavily criticized also by the base scientists. I'm quite optimistic to follow what is happening at the moment at the Miami project with Swan cells. They're going in very logical small steps from rodents to big models, even some primate models, now into humans. I think they've done now 5 as well. And they're actively enrolling again. It's in an early stage.
Is there any you would give your son if he had a spinal cord? Is there any you would give your son tomorrow if he had a spinal cord?
It's a very good question. There's any cell I would give my son tomorrow if he had a spinal cord injury. If my son tomorrow had a spinal cord injury, I would try to give whatever I think is the best I can allude to it in terms of neuroprotection. Because first you want to do what Brian just said a moment, you want to protect as much as you can in the acute stage after injury, when there is the lack of blood supply, when there is swelling, when there is inflammation, and additional damage of nervous tissue in the hours after the injury. And we have at the horizon a variety of drugs which have some promise.
So no stem cell you get?
Not yet, because I would wait what I can rescue there, and the stem cells that need to be given in an early protocol, let's say within a week after an injury, in my mind, are in the wrong place. They shouldn't be given then, because there's a considerable risk with such an intervention. You want to wait, and you want to tune these type of approaches to a subacute stage where you can assess the people, where you can follow them better, where you then can actually need smaller cohorts to be able they are efficient or not. So your answer is no, I would not yet do that. Absolutely correct.
I just wanted to expand on your answer a little bit, because one of the things that I get to do by working at the NIH is get a view of the entire landscape. And so I can address this a little bit, but one thing to start with is to think that when we move to clinical trials, it's a different path if we're moving a drug into clinical trials, a small molecule, or if we're moving a cell or a cell line into clinical trials, or if we're moving a device. There are three separate approval processes for good reason, because there's three separate types of issues that have to be dealt with. So with a drug therapy, very often we really have to understand where that drug goes, what the highest safe dose is, what the lowest effective dose is across a population, and really examine the toxicology across the whole body. With cell therapies that are going directly into the spinal cord, the issues are, do the cells survive? Do they tumorigen? Do they turn into tumors? Do they, if they don't survive, might they cause benefit anyway? What's the timing of putting them in? And the stage that a lot of stem cell trials are at right now, Neuron trial was ended for financial reasons, but the company transferred all of its rights to another company called Asterias, and they are reinitiating a new trial with brand new clinical data, 20,000 pages to the FDA of new clinical data. They're going to reinitiate that. But with a stem cell trial, we're still at the stage of, are the cells going to cause tumors? Are they going to survive? So would I give it to my son? No. But if he had an injury and he chose to be somebody to try something in controlled conditions in an FDA regulated and approved trial, I would be scared to death, but I would let him do it. That's where we're at with stem cells. We're not really, I don't think, at the point where we're looking at improved function. We're still trying to figure out what the stem cells do. With devices, there's one path if it's a device that's already approved for something else, which is what the epidural stimulator is. It's already approved as a treatment for chronic intractable pain. Nothing else will work. They'll put the stimulator in and it seems to alleviate chronic intractable pain. It was that stimulator that somewhat serendipitously enabled some movement. So that will then have to go through a long path. We'll have to figure out how it works, what it works, and optimize that. So I just want to emphasize there are three very different paths for these different possibilities, and they're at different places.
Your mic
I
over.
could, but
Do you want
I think
to?
we spent enough time on this one, so
Okay.
there's lots of other questions.
Okay, yeah. I have a quick question that Peggy can answer. Peggy, what's N?
So when us researchers say the word N, what that means is the number of samples that we're looking at. And so if we're talking about a clinical trial, it would be one person is an N of 1 and 5 people is an N of 5, or if we're talking about rats, an N of 25 rats, for example. So that clarifies it.
There's a nerd moment for you.
On the rat.
All right. Okay, you had your hand up one more time. Can you shout your question out and I'll repeat it?
So are these trials all done on people who have complete spinal cord injuries?
Good question. Can you
Are the trials that have been done, are they on people who have complete spinal cord injuries? Because I only ask that because I had an injury and mine was incomplete. So, and I, at this stage, almost 2 years after my accident, I'm getting things like, for example, I'll be walking down the hallway with some paralysis, and then all of a sudden my body feels like there's like a jerk, and then all of a sudden I'll do maybe 100 perfect steps where there's arms are moving perfectly and legs, and you'd never even know that I'd had an injury. And then all of a sudden, for no reason, it just collapses again, and then you could be walking like Quasimodo. So number one, is there any way to improve function for people who have only who've had not only but people who have had partial or incomplete spinal cord injuries? Number one, and number two, are they only doing any research on people who have complete spinal cord injuries?
Thanks, it's a really good question. I'm about to injure myself going over the water.
Wow.
That was on purpose. Alright, who wants to answer that question?
Please.
I think I gave them a heart attack.
Yeah.
Trials which we discussed today, they were done in motor, majority of them motor complete, but anatomically there were still some preserved fibers that had connections with the low level of injury, but they were functionally very not functional. But anatomical preservation of fibers still were present. That's why both Peggy and Chris showed that some anatomical connections still were present, but they were not functional. There are still clinical trials, not at this type of trials, going on with incomplete individuals like yours, locomotor training, and other trials available for people with incomplete injury like yours, but not probably for cell transplantations at the present time at least.
Well, I would just to comment that a lot of the experimental biological therapies and drugs in cells, particularly in cells, typically start in individuals with complete injuries. And the rationale there is that when the initial questions relate a lot to safety, that they would typically start asking those questions in individuals who have had complete spinal cord injury. Having said that, I think that the field generally feels that the therapies will probably have the most benefit for people with incomplete injury. That the therapeutic potential is probably greatest in individuals with incomplete injuries, those that have really functionally spared the neurological tissue going through their spinal cord injury that could be potentially the substrate for some of these treatments. It's just in the early stages, particularly for cells where there's some degree of invasive risk, that has started in complete. Having said that, the Asterios trial, that is the resurrection of the Geron embryonic stem cell technology trial, is proposed to actually go into individuals with incomplete injuries. And so the challenge there will be for them to prove that that it's safe, that the injection itself will not cause damage. And I think that anybody with incomplete injury, well, anybody with a spinal cord injury, but anybody with incomplete injury, you really treasure what you got. And the thought that you would potentially lose some of what you fought so hard to recover and retrain, rehab, is really quite daunting. So, but I think your question around, are these therapies going to be effective for individuals with incomplete injuries? I think that conceptually, I think people feel like that's probably the most likely kind of individual to gain from these. It's just that's not how a lot of the more invasive things are started.
Yeah, same with the drug trials for neuroprotection. If you have a complete injury, and essentially most of it to corpus damage or all of damage, the chances to rescue is very little. So again, the likelihood of seeing some improvements is in the incompetence.
Yeah.
This being said, they're the hardest group to study because they have a lot of spontaneous recovery. There you have to tease out the cohorts that would improve themselves to a certain degree from the ones that are now getting even better because they have now received the drug. And here we see the big dilemma. It takes, in that case, 100 and more patients, 200, 250 patients, to be able to tease out an effect in the cervical incomplete injury that drug A or B that needs to be given within 12 hours we have. And that's a huge challenge.
Thank you. We have another comment online and then
So, invariably, I think this question comes up at what do you envision in a cure for spinal cord injury, and how far back are we from that cure? What do you envision in a cure for spinal cord injury, and how far back are we?
the big kind of a blue sky question. So predictions, thoughts on what gets us there, etc.
In general or just specific? Because in general it's protecting first as much as you can.
Are you answering the question now?
Please answer. Okay. I can give a very general answer. Initially, protection as much as you can in the early hours, days after injury with a neuroprotective, probably drug treatment, compound treatment. This usually goes hand in hand with the surgeons stabilizing the column so you don't get further compression injury and other additional injury due to instability. Then assessing what is left, and eventually down the road, decade or longer, assess what type of cell may be the most suitable one. Is it a cell that is providing insulators for the remaining cables, as they may have lost the insulators, like a copper cable or using the plastic? Do we have to put more plastic around the copper wires? Or do we need little cells that are now producing relays, and like telephone relays are receiving inputs or sending fibers further down again and circuiting them with appropriate rehab into function and coercing them? Those are questions which then become very individual, and it becomes almost like a personalized approach. While the initial approach of protection may be probably more a general one, and even there, we just discussed 2 hours ago, even there, some patients may lend themselves for certain type of neuroprotection than others, because different neuroprotective candidates you see in animals have different properties. Some are more targeting blood vessels, others more inflammation, others more the hyperexcitability of nerve cells. So it's complex, but it will be a combinatorial approach.
I sort of have a add-on question to that. So when you say a combinatorial approach, why not just one cure? What is it that means that we need many different kinds of cure?
Well, I spoke long enough, I think, because they have different levels, different severities, different type of injuries, which lend themselves automatically to different parts, which are disjointed, needs to be restored in different functions. And I see Brian itching there, he wants to add something. Hungry. I pass it on at this moment.
I can try. So why are we expecting to be a combination?
Well, the overall question is
when is the cure?
What's going to get us there, and it's with the atom, and why do we keep talking about combinatorial?
Right. So, I work in an institute where we study a lot of neurological disorders. And that question comes up for every neurological disorder, because the cure is regaining what you don't have now. And it's a constant moving target until you are back to where you were before you were injured. So I think cure is a stepwise process. I also think all of the things that have to be fixed are very different. And in part because our regulatory agencies want to see us show one thing does one thing well, and in part because the nervous system is so complicated to begin with. When you developed as a baby, you didn't develop with just one signal. You developed with a lot of signals, a lot of cells, a lot of coordination, and a lot of exercise and training. So I think that
You know, it is a stepwise progression. I think that already some of the things that are happening in rehabilitation are improving the extent to which people are recovering from injuries. There's fewer who recover nothing. But that's not very heartening to those in the audience or those that you know that may have recovered nothing. But we are moving in the right direction. But cure is a moving target.
Well, I think that you know, we get asked a lot about the issue of cure, and I think probably our idea around the concept of cure perhaps a bit incongruous with what I think a lot of people think is cure. Because if we visualize something that is going to restore somebody to full physical function, you know, as the outcome of the treatment, I think that we're a long ways away from that. I think that the things that we realized in studying the complexity of spinal cord injury is that indeed there's lots of, you know, aspects of the injury that need to be addressed in combinatorial fashions, in different manners. And I think that conceptually, the idea that, you know, we are constantly inching the ball forward, down with the ball. There are no field goals. There are going to be no Hail Mary probably passes. It's going to be inching the ball forward. And I think that's, you know, when you look at some of the things that come out, you know, they represent, you know, moments in our field that we've maybe inched the ball forward. And I think that is how, you know, we look at, if we kind of view cures to mean things that will incrementally improve the lives of individuals with spinal cord injuries, then that process is going to occur in small increments with small victories, which is, I think, a different vision than what I think a lot of people have in terms of, you know, what a cure represents. But I think that those of us that study this, you know, that's most likely how it's going to roll out. And then on the issue of timing, I think that this is something that comes up a lot, because we do get asked, and my patients will ask me, like, well, what's going on? Like, why is this taking so long? And I think that, you know, as an orthopedic surgeon, one of the things that I reflect back on is we all understand that nervous tissue in the central nervous system doesn't really repair naturally the way that, you know, and that's evidenced by the permanent functional loss of individual spinal cord injury experience. There's a system in the body that actually heals quite well, and as an orthopedic surgeon, we take advantage of that a lot, and that's bone. Bone tends to heal quite well. Bone is actually one of the tissues in our body that actually heals remarkably well, and even in some respects, even without any scarring. And people have been studying bone biology and bone healing for a long time too. In the early 60s, there was a guy named Marshall Urist who identified the first time one of the proteins that mediated bone healing, bone formation. He published that in a very prestigious journal called Science in 1962. And that led to a lot of research around what was actually inducing bone formation, how bones were actually formed. And a lot of people were studying this. And this is in a system that naturally heals. The first FDA-approved product to receive approval for commercial use in the concurrent of bone healing was approved in 2004. And this is in a system that, in a biological system that actually heals on its own. So when you put that in the context of a system as complicated as spinal cord injuries and neurological injuries, you know, what we see is that, you know, we're facing a very complex problem, and that progress is going to take these small wins, and they're important. But I think that the idea that it's going to be, you know, a Hail Mary 40-yard pass down the field is, I think, probably not what's going to happen.
Okay. Sorry. I know there's quite a few more questions left, so I don't want to miss out. So I know I said Dina first, and then Peggy next.
I have a very simple question considering the ongoing panel. And that's kind of when you, how, this little paper that you guys gave up here kind of talks about a time frame of 1 to 10 years. But how long after a clinical trial before some of these breakthroughs hit the public healthcare system and actually kind of make a difference to this community who is actually affected by spinal cord? And then I have a part B that might be more challenging for the master of the panel.
Okay, we'll try and get back to that part B. So the question is, how long after a clinical trial concludes do things get into the healthcare system to be used by patients or doctors? Anybody?
I have to say, it was quite shocking when the first results from the master's trial came out and that everyone had a very mild trend, and it was essentially overnight happening that doctors were becoming sued by lawyers who were telling their patients, if you haven't gotten that already now, there's the others out there to get it now. And it was happening very quickly for fear of litigation. So it's really depends on this. And it was quite, in those days, it was frustrating situation for the surgeons because they didn't have enough time to get the information through their own associations and professional collegial colleges to actually digest the information of the trial and decide, yes, this should be now our clinical practice. It was actually very, very quick. So it depends really on the case, what it is, and whether there is litigation suddenly enforcing the issue.
Yeah, I think that to some extent the answer to your question is not really known because we haven't completed a lot of clinical trials in spinal. We'll talk about the methylprednisolone example, and that certainly that demonstrated that the uptake of that therapy was almost overnight. And so, you know, I think the thing that would be a bit harder to estimate is if the therapy, such as a embryonic stem cell, a neural stem cell, is shown to be efficacious, what that will mean in terms of you know, the translation of that into the healthcare system. You know, there was a drug or there was a treatment that was being evaluated. It was a monoclonal antibody. It was administered intrathecally, which was the really the combination of probably the most extensive preclinical experiments. Oh, I'm sorry. They put it into the spinal cord, basically, into the fluid around the spinal cord. And that study, I think they were looking at a potential cost of some $300,000 per patient that ever got through. So there are issues like that that will complicate you know, the uptake. And unfortunately, because we haven't had a lot of clinical trials actually get to that stage, we don't know the answer to some of that.
Is that disheartening for you, or do you feel like there's barriers in place? For example, the information isn't disseminated properly, or even if the minimal kind of trials that are available, have they, like, what is the big barrier? Financial, or what is the?
Well, certainly, I think the big barrier is actually in the evaluation process. And I think Geron actually provides a very good example of that. So, on January 19th, 2009, Geron received an FDA approval, and their stock went up about 10 times. So a lot of excitement to do the trial. And they, you know, they spent the next 2 to 3 years kind of getting the trial going, getting all regulatory approvals, and then really it shut down after 5 patients got treated. And so now here we are in 2015, so 5 years and change after that very dramatic announcement. And Asterios is talking about a trial that is probably going to take another 5 to 10 years before we'll know really what whether they're safe and maybe even some sense of whether it's effective. So the challenge is actually, I think, to some extent in the evaluation process. And that's, to some extent, is often overlooked, the fact that the actual process of evaluating these technologies is so long, which then leads us to the challenge of deciding what it is that we should be putting into clinical trial, because it is such a prolonged and extensive endeavor.
It's sort of a related question. It's written down, so I don't know who asked it. If you want to claim it, you can raise your hand if you want. Is for-profit research likely to hijack SCI research and make it proprietary technology, i.e. very expensive and restricted in scope? Like, what's the role of corporate or commercialized science? Anybody?
My initial response will be, if you want to make money, don't try spinal cord injury.
I'm happy to make a comment on that. So I think that this is a complex issue in our field, and it pertains really to the relationship between academic investigators and universities, both of them, and corporate entities that have the ability to take a technology forward and put it in a clinical trial. I think that it is important to recognize that industry and commercial entities have a role in taking these technologies forward. And I think people could argue that in a perfect world, everything would be done by academia. But I think that we realize now that the cost of things that, in fact, industrial partners, commercial partners are important for taking those nascent ideas, those promising ideas out of the academic lab and really bringing them forward through a very complex process that is, to some extent, foreign for academic individuals. And I have to admit, I was, when I started my scientific research career, I was involved in the development of a drug, and that drug was commercialized, and I had nothing, I had no intellectual property, I had no patent interest in this product. But I was very interested in seeing the process of what it would take to bring it forward. And it shocked me, the process of actually taking that drug, preparing an FDA submission, going to the FDA to talk about that. It's something that's actually totally foreign to what academic researchers actually understand, and was totally new to me. And so, academic or industrial commercial partners are actually a very important part of the process. And I think that the concerns around the integrity of the research that people raise, those are important questions to ask. And I think that as a community are grappling with that, or understanding how to grapple with that issue better. But I think that it is important to recognize that industrial partners, commercial partners do actually have a role in taking those promising things from the scientific labs and actually navigating them through this very complex process so that they actually can be tested in human patients.
Just to add, it takes a lot of money, and oftentimes to raise this money, companies have the know-how how to go with that concept to a stock market and raise more money up. And I would say what we really need more is to actually be companies and academic scientists that help companies and advise them in doing the right experiments and getting it right so that they are not wasting their time and that we are exploiting their capacity in a good way and building trust between academia as independent collaborators and consultants to them, and then as the ones that can push things over the finish line and finally get things to trial.
So we're getting very close to the end of the night, full bellies, and we're all tired. And so we have one more question from online, and then we're gonna do the wrap-up process and some surveys and a door prize, so don't leave yet. But I think we've left some contact information. If you have a question unanswered, you can email us, and we will make sure it gets answered to the best of our abilities. And here's one last question.
Considering the few available subjects for human studies, how should we deal with the variation in treatment responses that we observe in different subjects?
Wow.
Considering the few available subjects for human studies, how should we deal with the variation in treatment responses that we observe in different subjects?
I kind of wonder if that person has a master's. Maybe they're just a research here.
You probably heard the wow from here panel of experts. It's a very, very good question. One of the criteria which we're using to include patients into the clinical trial, we're trying to definitely select patients as close as possible with respect of their severity, levels of injuries, and patients will be homogeneous with respect of their severities and so on and so on. It's one of the criteria in order to improve the end of patients, number of patients in the clinical trials. Scientists and clinicians around the world clearly understood that we cannot do any more trials in one single center. And this has created very good understanding around the world that clinical trial studies should be done on collaborative basis. That there are numerous, right now, clinical trials done either through the multi-center studies within one country or multi-center studies across the world. There are numerous centers or initiatives that provide even data-based how we can organize and include subjects who are interested in clinical trials and so on and so on. That's only this way how we can find patients for these kind of clinical trials around the world or within the countries. This is only one way how we can then find the best way fitted patients and specifically for the specific studies.
Well, I think that's that's a fantastic question, and really underscores one of the major challenges we have in developing therapeutics. Is that we're actually now, I think, beginning to grasp this concept that that the heterogeneity of injury is such that, as the person who asked the question, leads to all these different responses. Maybe that was totally obvious to individuals that have a spinal cord injury that look, everybody's different and we have different responses. And we unfortunately currently stratify and classify in a very crude manner, and we don't currently have great ways of discerning how individuals are different and how they may respond differently to different therapies. So I think a lot of research that may seem actually kind of boring to people is related to trying to understand those differences so that we can predict, we can understand and then predict who is going to be responsive to certain things, or who has a better chance of being responsive to certain drugs, and who's going to have the chance to recover differently, so that we get away from, as Chris and Peggy have shown, this classification of A, B, and C, and D. We obviously are missing a lot of the complexity of the injury in that kind of classification. So we are trying to recognize that there is this variability, trying to understand that at a more biological level, and hopefully that will enable us to test therapies in a more efficient and effective manner.
if I can do just a real quick follow-up to that, there's, in a very optimistic voice, it is a huge problem, but there are projects ongoing internationally and at the NIH and at Canadian Health Institute to better define outcome measures that have a sliding scale of improvement that can better be used across different types of injuries a little bit. We're not there yet in terms of defining something that's going to be a really good measure on a sliding scale, but hopefully that's the goal, is to be able to develop an outcome measure that will allow clinical trials to take on a wider variety of people, and then we don't have to pick and choose quite so much.
Yeah, just very quick, as we go on, the fields of pharmacogenomics is increasingly identifying genetic makeups of some human beings responding to drugs and other human beings not responding to drugs. And eventually that information can be factored in to then exclude those that turn out to be non-responders and be able to sort out through the noise.
So that's a great note to end on, because you know, there's so many different kinds of spinal cord injury, and I think we've talked about many times tonight that it takes a lot of different types of people to contribute to spinal cord injury research. And I was just struck by the fact that, probably by design, I mean, I wasn't one of the organizers, but we have a basic scientist, we have a research funder, and you're a basic scientist, weren't you?
Was.
A clinician, scientist, surgeon, another clinician scientist, and a community partner. Also, you're a basic scientist before. So that just shows you that's just 5 of the different sort of flavors of researchers in spinal cord injury. And if you ever come over to ICORD, you'll see, you know, probably 5, 10 more flavors, in addition to all the trainees that sort of make the research labs go. So, so we hope that tonight we've given you an opportunity to ask some questions and to hear a bit more about what goes on behind the headlines. But on that note, I just want to thank our experts for coming and for sitting through tonight and at the end of your long day. So can we give them all a hand? And we'd like to thank you all for coming and for taking time out of your day to hear about research, which is not always on everyone's top of everyone's radar. And we'd like to thank GF Strong for having us, the Canadian Institutes of Health Research for sponsoring the event, and that's also a research funder that provides funding to researchers to hold public events like this one, as well as Spinal Cord Injury BC, Moriah Restaurant, Lonsdale Rentals, and ICORD, the International Collaboration on Repair Discoveries. And remember, you can always get answers to your questions about SCI research, or at least we'll do our best to answer them by emailing admin@icord.org. And for everyone here in person, there's a survey on the table in front of you. Please, please, please fill the survey out. It's something CIHR asks us to do, and it helps get funding like this for more public events happening. And if you came without registering and you want to hear about the next event, make sure to leave your contact information on the survey form. And you can put your completed surveys in Jess's basket over there. And thank you so much to the people who attended online. I know it's not the easiest thing in the world, and we really appreciate you coming in. And there's going to be a door prize draw. What do I do? Okay, give your survey to Jess, and there'll be a door prize. And just a shout out to Leanne and Jack, who are the organizers of this event, who did a great job. And our trainee volunteers who've served us food so well. Thank you. The email for questions is admin, ADMIN, @icord.org.